Hi, my name is Harper. My name is Elizabeth, and this is the process of the Wolbachia project in our Biology 1107 lab. A little bit on the biology of Wolbachia. Wolbachia is an intercellular parasite that is found in arthropods. Insects and Wolbachia have a parasitic relationship. Wolbachia infects nearly 20% of the planet's arthropods, and Wolbachia is also considered the most common endosymbiont in the world. This is significant because it increases the female population through 4 different phenotypes: male killing, feminization, parthenogenesis, and cytoplasmic incompatibility. All 4 of these either cause males to not develop or to die. And the relevance of Wolbachia to human health we see in viruses like dengue fever and Zika fever that the animal vector and Wolbachia host are both a mosquito, and Wolbachia and the mosquito have a parasitic relationship. Wolbachia essentially shortens the mosquito's lifespan, which lessens the disease transfer. And something important to note here is that Wolbachia bacteria does not directly infect humans. 2 different diseases caused by the Wolbachia is river blindness and elephantitis. The animal vectors are the black fly and the mosquito. Both of the hosts are the filarial nematode, and they have a mutualistic relationship. Therefore, if you kill the Wolbachia, then you kill the nematode and the disease. Now, a little bit on the importance of the Wolbachia project. Why are Georgia Southern Armstrong students participating? Well, we have a limited number of devoted Wolbachia scientists. It also gives a hands-on experience with the process and application of science. And why is individual experience important? It's important to practice DNA extraction, polymerase chain reaction, gel electrophoresis, DNA sequencing, and bioinformatics skills. And most importantly is to answer the research question, is my insect infected with Wolbachia? So throughout the project, we followed the steps of insect collection, insect identification, searching literature, and constructing a prediction. We then extracted the DNA, performed PCR on the DNA, did gel electrophoresis, evaluated the results and the prediction from those results, and then DNA sequencing. On this next slide, we have information and methods used to collect our insects. We collected our insects on the 24th of January, 2024. We collected them around 1 p.m. and we collected them in the grassy area outside of Armstrong campus. It was very overcast and humid and warm and damp when we collected, and we mainly used a sweep net, but we also used forceps to transfer our insect specimens into the jar. So this is an image of my insect order Diptera. As you can see, it has 1 pair of fully developed wings, and then the hind wings are reduced to tiny knobs. I found this insect order by using the dichotomous key steps 1 and 2. And my insect order is order Hemiptera, and this order has hard, dark outer wings and then transparent thin inner wings. It also has a needle-like mouth part, and I use dichotomous key steps 1, 2, 3, and 4 to find this. A little information about the insect order Diptera is that it lives in freshwater. They're semi-aquatic in most terrestrial environments worldwide. Their diet consists of honeydew, nectar, exudates, and blood. The life cycle is egg, larva, pupa, and adult, and then a unique trait is that some Diptera antennae act as their speedometer. And some information on order Hemiptera is that they are mostly aquatic insects, and they have needle-like mouth parts, which means that they feed off of a liquid diet. And their life cycle lasts about a month, and they go through egg, nymph, and adult stages. And one unique trait about this order is that they have an outer hard pair of wings and then an inner thin pair of wings. So through my scientific literature review, I've found 2 journal articles that have a prevalence of 90.8% and 17.98%, and then the local information stated that there was a prevalence of 18.5% of insects captured. And for my order Hemiptera, the journal articles that I found stated a prevalence of 20.3% and 67.8%, and then the local information provided was only 25%. My prediction stated that if I test the DNA extracted from my insect specimen with molecular biology techniques, then I will not find a presence of Wolbachia. This was because all 3 of the prevalences added up to be 42.43% average, which is less than 50% chance of finding Wolbachia in a sample. And my prediction was that if I test my insect specimen using PCR, then I will find it to not be infected with Wolbachia. And I also decided this because all of the data concluded that there was only 50%. So in exercise 5, we did DNA extraction from both of our specimens, and we found good quality DNA. You find the quality of the DNA by testing the A260-A280 ratio, and as you can see, both of our ratios resulted in higher than 1.7, resulting in good quality. Good quality DNA means that there is a low amount of protein to contaminate the sample. And our expected PCR results, we both expected negative for Wolbachia gene and positive for the insect gene. This is an image of our gel electrophoresis. We used a 2% agarose gel with 100 volts. This first lane shows our positive control, which shows the Wolbachia gene and the insect gene. The negative control only shows the insect gene. There's a blank, no template control, and then both of our samples show the insect gene but no Wolbachia gene. And my prediction was supported both by the gel electrophoresis and the scientific literature review that we did.
My prediction was also supported by the scientific literature and the gel electrophoresis.
So my individual benefit from this was before the lab, I was not able to pipette, extract DNA, or perform gel electrophoresis. Pipetting is used in several lab exercises and real-life lab applications. The DNA extraction taught me to use PCR and identify good quality DNA, and from gel electrophoresis, I learned to analyze collected DNA and test my prediction.
And some individual benefits that I got from the Wolbachia lab experience was an enhanced understanding of the significance of Wolbachia in insect populations. I also gained proficiency in techniques such as pipetting, PCR, and gel electrophoresis. I also learned proficiency in using software such as OrFinder and Blast.
For all of you interested, here are our references, and don't forget to like and subscribe down below.